Cell invasion, in which cells pass through the thick extracellular matrix of basement membranes to enter neighbouring tissues, is crucial for both normal development and for the metastatic spread of cancer cells. But relatively little is known about the process because it is hard to image in vivo, at least in vertebrates. David Sherwood from Duke University, however, studies cell invasion in C. elegans, where, during hermaphrodite development, a single uterine cell called the anchor cell breaks through basement membrane and attaches to the underlying vulval epithelium.
Anchor cell invasion is a highly stereotyped invasion event. It happens over a very precise 90-minute window in the L3 larval stage of C. elegans. Furthermore, we can easily visualize it. It's only about 20 microns into the worm. And on top of all of that, we also have the genetics. We can do genetic screens as well as whole genome RNAi screens. And so for that reason, it's just a very powerful model. And in particular, what we wanted to ask in this paper is to very carefully look with high-resolution imaging, how does this cell get through the basement membrane?
Sherwood and colleagues, led by grad student Elliot Hagedorn, began by imaging the actin cytoskeleton of anchor cells before invasion began, revealing the presence of small actin puncta near the underlying basement membrane.
They're very dynamic. They turn over every 45 seconds, and there are a lot of them. There are hundreds of them that turn over incredibly rapidly.
Cancer cells invade extracellular matrix in vitro by forming actin-rich membrane protrusions called invadopodia. Like invadopodia, the actin foci in anchor cells were protrusive structures that contained actin regulators like ENA-VASP, PIP2 and RAC-GTPases, and they were inhibited in the absence of integrin cell adhesion molecules. This suggests that, in vivo, anchor cells initiate invasion by forming invadopodia-like structures.
What we see is one or maybe two of them breach the basement membrane. And so it's not like a shotgun blast where you have a bunch of holes punched through the basement membrane, but just only one or two of them go. And for the one or two of them that go, then one of them forms a very large protrusion that widens just a single gap in the basement membrane and directs an invasive protrusion between the central vulval cells. And when this protrusion forms, the invadopodia that are remaining in the anchor cell disappear. So it looked like there was a transition occurring.
Hagedorn et al. Wondered whether proteins required for anchor cell invasion might help regulate this transition. In particular, they examined the role of the Netrin receptor, known as UNC40 in worms, which localizes to the interface between the anchor cell and the basement membrane.
Initially, it seems pretty spread out along the entire invasive cell membrane of the anchor cell, and it's not co-localized with the invadopodia, and its loss doesn't affect the invadopodia. But right at the point of that initial breach, the receptor concentrates at that point and further concentrates as that first breach opens up. And what we saw was that in a Netrin receptor mutant, the initial breach was delayed. The invadopodia continued to form. They didn't shut down. And that no invasive protrusion was formed. So the Netrin receptor is somehow either initiating the first breach, or, and what I favor, is that this receptor somehow is detecting that initial breach in the basement membrane and reinforcing it.
Mutations in the Netrin ligand, which is secreted from the nearby ventral nerve cord, cause similar defects in anchor cell invasion, though the phenotype wasn't as severe, with the Netrin receptor still concentrating at basement membrane breaches. Downstream, the receptor recruited the actin regulators ENA-VASP and RAC to the breach site, stimulating F-actin assembly to promote the growth of a stable invasive protrusion.
The receptors are recruiting these F-actin effectors that would usually be dedicated to the invadopodia to this invasive protrusion. It's basically acting as a sink, both generating the invasive protrusion that invades the vulval cells, thus mediating uterine vulval attachment, and shutting down additional invadopodia so that you only get a limited number of breaches in the basement membrane.
Cell invasion is generally thought to be driven by proteases that degrade the extracellular matrix. But Hagedorn et al. Thought that anchor cells might widen the basement membrane gap by physical means as well.
When we were looking at these anchor cells invading, we were really struck by the accumulation of laminin and type 4 collagen on the edges of the basement membrane gap as it was widening. And this suggested to us that maybe this anchor cell with this protrusion going through is in part pushing the basement membrane aside through a physical displacement.
However, the accumulation of matrix proteins could also be due to the secretion of newly synthesized laminin and type 4 collagen. To distinguish between these possibilities, Hagedorn et al. Tagged the matrix components with a photoconvertible fluorescent protein called Dendra.
And we simply converted the basement membrane from green to red directly under the anchor cell just before it invades. And every single time, like the parting of the Red Sea, we got the parting of the basement membrane, showing that actually physical displacement is involved. And that was a big surprise to us, because this was not thought to be possible. The basement membrane is supposed to be tightly cross-linked. It's, really tough. So now we're really interested in how that works. We're looking at, for example, cross-linking enzymes and seeing are they differentially expressed. We're really interested in looking at the forces on the basement membrane, and then finally looking at proteases, because one of the things that proteases you could imagine doing is softening up the matrix by creating clips in it, allowing it to be pushed aside or displaced more easily.
Sherwood and colleagues are also interested in examining whether cells use similar mechanisms to invade basement membranes during vertebrate development and cancer metastasis.
Basement membranes go back to the dawn of multicellularity. They may have been required for multicellular life, for animals anyways. And so, in my mind, cellular programs to remodel, to break through basement membranes are likely to be ancient and conserved. And so I think that we'll find many commonalities between what we find in the anchor cell and what's found in metastatic cancer cells, as well as normal vertebrate cell invasion events.
In the meantime, you can learn more about how the Netrin receptor focuses invadopodia-driven basement membrane transmigration in the paper by Hagedorn et al., published in the June 10, 2013 issue of the Journal of Cell Biology.